Good evening, everyone. Welcome to Cell Talk presentation. For today's Cell Talk presentation, Vijay is our presenter and he will be discussing on the topic a faster, non-invasive artificial intelligence olfactory-like mesh system that aids diagnosis of Parkinson's disease. Before starting the presentation, let me introduce Vijay. Vijay Padul is working as scientist bioinformatics at Renix Life Sciences. At Renix, he is leading multiple research projects on bioinformatics, genomics, and next generation sequencing research related activities in the organization. His current research projects include the development of personalized neoantigen cancer vaccines and the utilization of publicly available multi-omics cancer datasets to create new insights into cancer biology and its application in cancer therapy. Vijay has completed his PhD in life sciences from Homi Bhabha National Institute, Mumbai. His PhD study focused on cancer genomics involving multi-omics analysis of glioma brain tumor subtypes. He has over 10 years of experience in the wet lab as well as in silico approaches for multi-omics studies, which include exome sequencing, whole genome sequencing, transcriptome sequencing, and analysis. Previously, Vijay has worked at various graduate and postgraduate degree colleges as an assistant professor. Thus, he brings multi-omics expertise and teaching experience to the Omics 5 team. At Renix, Vijay has established bioinformatics pipelines for personalized cancer genomic analysis, and his research has highlighted the importance of molecular marker analysis in personalized cancer therapeutics to match the treatment strategies to patient cancer tissue molecular characteristics. His contribution include assessment of patient HLA genotype status for tailoring the personalized neoantigen peptide therapy and glioblastoma subtype analysis. His larger aim is to utilize personalized genomics approaches for aiding effective personalized treatment strategies for cancer patients. Now I welcome Vijay to start the presentation.
Thank you, Uma, for the introduction. So, hello everyone. For today's Cell Talk, I have chosen a research paper titled "A Fast and Non-Invasive Artificial Intelligence Olfactory-like System That Aids Diagnosis of Parkinson's Disease." The original authors are Gina Cao et al. The topic selected for the presentation is Artificial Intelligence in Neuroscience. The title for the presentation would be Innovative Olfactory Diagnostics and Artificial Intelligence Application to Diagnose Parkinson's Disease. The journal in which this paper is published is European Journal of Neurology with impact factor 3.9 and the year of publication is 2023. The research presented in this paper has impact on Parkinson's disease. So, Parkinson's disease is a neurodegenerative disorder which is affecting older people, and as the age increases, the severity of the Parkinson's disease increases, and this disease is affecting approximately 1% of the elderly. And aging is the principal factor that drives this disorder and neurological degeneration. The diagnosis of Parkinson's disease is evolving from clinical and biomarkers supported methods. The presentation of this disease is the motor symptoms that include initiation of tremors and inability to support the person's gait, and also other motor disabilities to carry out the daily functions essential for the doing daily activities. But in addition to that, accurate diagnosis of the different stages of the Parkinson's disease and subtypes is challenging right now for the clinicians. In addition to the motor symptoms that we have seen before, there are non-motor symptoms also, which have been associated with the initiation and progression of Parkinson's disease. And this, which have been recently identified, these include olfactory dysfunction and also skin-related disorders such as seborrheic dermatitis, which has been strongly associated with the diagnosis of Parkinson's disease. At most like 50% of the patients, they are having this associated disorder, that is skin-related disorders, and specifically seborrheic dermatitis. And therefore, an interest has arisen in sebum, which is integral factor in seborrheic dermatitis. So therefore, this chemical that is secreted by our skin, which contains different organic molecules. And the sebum, it has been an underexplored biofluid and therefore there is a possibility that there could be potential biomarkers which may be useful for diagnosis of Parkinson's disease and also to study the progression of Parkinson's disease, as it is strongly related to the Parkinson's disease diagnosis and its involvement in seborrheic dermatitis. There is also a hypothesis that the patients who suffer from Parkinson's disease, they exhibit abnormal odor, and which may be due to abnormal sebaceous gland secretion. And therefore, this interesting hypothesis is worth uh, pursuing to distinguish whether uh, Parkinson's disease patients indeed smell different from healthy individuals, which are aging in a healthy manner. And if we can find this certain distinguishing factors in the sebum or any odor or olfactory distinguishment between the Parkinson's disease and the healthy controls, we can utilize these differences as a biomarker for diagnosing Parkinson's disease and also to study how Parkinson's disease is progressing and whether we can track that progression of Parkinson's disease by using these olfactory markers. So sebum is primarily secreted by our skin, and it contains multiple chemical components, which are cholesterol, triglycerides, free fatty acids, squalene, and waxy esters, uh, along with the components of our microbiome and their microbial activity. In 2021, Sinclair et al. They had confirmed the presence of differential regulation of volatile organic compounds in the sebum of Parkinson's disease patients. In that study, they had used gas chromatography and mass spectrometry, and also liquid chromatography and mass spectrometry analysis in their study. So, if indeed there are any differences between the Parkinson's disease patient and healthy controls, then it is worth for its application in the clinical setting. And therefore, in this particular study, they have described an artificial intelligence olfactory-like system, and they have developed this system, and which can be used for screening of potential Parkinson's disease patients and their diagnosis. And this system has already been utilized for diagnosis of the respiratory diseases and digestive diseases. The same application can be extended to the Parkinson's disease patients also. So this artificial intelligence olfactory-like system, it works by detecting odors from sample and that contact the active material of the sensor. What they do is they extract the odors or the secretions of the screen, and once that sample is collected, they treat that sample and utilize that sample in a way that different sensors can identify different biochemical components of that odor. And depending upon the composition of that odor or the skin excretions, uh, then machine learning algorithms can be utilized to identify whether that sample belongs to a healthy individual or a diseased individual. So in that workflow, the response that get from multiple different sensors, that it is converted into an electrical signal, and that electrical signal is used as a base data for machine learning based training of the models, and also to analyze different odors based on the differences that arise in the electrical signal based on the background or the underlying chemical compositions of the skin secretions or the odors. Right now, the present challenges that are there in this particular area is that accurate diagnosis of the Parkinson's disease, it remains a challenge for the clinician. Many times Parkinson's diagnosis may be misdiagnosed, or it may be diagnosed late at advanced stage. And therefore, it is urgently necessary to develop reliable diagnostics and also prognostic biomarkers for the diagnosis of the Parkinson's disease, so that the initiation of Parkinson's disease can be identified correctly and proper remedial measures can be taken, so that progression of this disease can be arrested at the initial stages only. This research article had the objective to develop a fast and simple artificial intelligence olfactory-like system for rapid determination of sebum biomarkers from Parkinson's disease patients to provide a rapid, non-invasive and inexpensive diagnostic tool for the Parkinson's disease. If this is possible, then it will be really helpful to diagnose the Parkinson's disease. For that, they only need to get the sebum sample from the patient sample, which is non-invasive process. Simple skin swab can be utilized for this particular purpose. And as it is just based on the analysis of the sebum components, so therefore it will be a rapid method, and depending upon the correlation that is found between the differences in the healthy and the Parkinson's disease patients' sebum samples, it will be an inexpensive diagnostic tool for accurate diagnosis of the Parkinson's disease. So this slide describes the study design of this particular research article. So they recruited multiple patients, around 200 patients were recruited for the study, and from their upper back in a controlled environmental condition, they collect the swab of the skin of the upper back by remaining the swab in contact with the skin for 10 minutes. During that time, whatever sebum or the skin secretions are there, these are absorbed by the swab, and then these skin secretions that are absorbed by this swab, these are collected and they are treated to the gas chromatography like system, in which various components of this sebum, these are treated to this detector, multiple arrays of detectors, which are specifically able to detect different components of this sebum, and that array of sensors, they create an electrical signal which are converted into an response curve by SAW sensor, and between the healthy individuals and the Parkinson's disease individual, these response curves, these are compared, and the differences and similarities between these are identified by analyzing this data by using different machine learning algorithms. They have used specifically 3 machine learning algorithms, and depending upon the correlation that is established, this analysis is able to distinguish between Parkinson's disease patients and healthy control. So this is the description of the study participants. So the study population comprised of 250 participants, in which 121 were Parkinson's disease patients, and the rest, that's 129, were control subjects. Out of these 121 Parkinson's disease patients, 3 were drug-naive patients. One limitation of this study that has been found that these Parkinson's disease patients, they were already on medication. So therefore, there is bound to be an impact of medication on the Parkinson's disease progression and the symptoms that may be apparent in the sebum composition of the patient. So this is one limitation that has been acknowledged in this particular paper. As there were only 3 drug-naive patients, that means the Parkinson's disease patients in which they have never got any drug. So as the population of this particular group was very low, so therefore comparison of this group with the other group was not possible. So they have continued this Parkinson's disease patient comparison with the 129 control sample, despite the Parkinson's disease patients were under the drug treatment. So this is the comparison of the 2 groups, Parkinson's disease patient and healthy controls. So as you can see, the groups are fairly balanced in these 2 groups in terms of gender, male, female, also mean age, body mass index, disease duration, then UPDR3 score, and also Hoehn-Yahr score, and but only in this case, drug-naive patients were only 2.5% in the Parkinson's disease patient. For sample collection, they had set certain conditions in order to bring uniformity into the collected sample. So each participant was required to comply with the following conditions for the 12 hours prior to sample collection. That they should not bathe or use body lotion up to 12 hours before the sample collection, and they should be refraining from strenuous exercise, because exercise might induce more sweat production and therefore might change the sebum chemical composition. And also they should avoid the use of nicotine, alcohol, or perfume. which might add some other chemicals into the sebum excretion, and therefore will not be representative of the natural sebum excretion of the patient's skin. To collect the sebum sample, the participants were swabbed with sterile cotton-based medical gauze, which was of the size of 7.5 centimeter to 7.5 centimeter, and the sample was collected from upper back of the patient, and the environment of the patients, it was kept at 26 degrees Celsius, so that uniformly all the patients would be in the same temperature conditions, so that the same amount of sebum may be generated, and the effect of temperature will be nullified. This gauze was kept in contact with the skin surface for 10 minutes before the sebum was extracted. Then the gauze was kept in a inert plastic bottle, and gauze was transported to Zhejiang University, and it was stored at minus 25 degrees Celsius until the analysis of the sample was carried out. For further analysis, a sensor which is called a surface acoustic wave sensor, it was used to detect the mass loading of the compounds separated by gas chromatography. There were 3 modules in the artificial intelligence olfactory-like system which they have developed, and these 3 modules included the first module was gas injection and pre-concentration stage, then chromatographic separation stage, and after that sensor detection, in which multiple arrays of different sensors were utilized, which were specific for different components of the sebum. Then the volatile organic compounds that were present in the sebum, its data was output from the artificial intelligence olfactory-like system, and it was recorded in the data set. In the data analysis, the Parkinson's disease and healthy control samples, they were divided into training and testing data sets into 8:2 ratio for the machine learning methods. 3 classic machine learning models were used. First was the random forest, extreme gradient boosting, the second, and the third was gradient boosting decision tree model. For the model to distinguish the severity of Parkinson's disease, patients were divided into early stage, middle, late stage, and these stages were according to the Cohen-Yar classification. So in the second stage, they tried to study the differences that might arise into the sebum due to the progression of the disease. So here, this slide, it shows the result of the analysis, and here they are showing the average frequency of the volatile organic compounds from the sebum, and the comparison in 2 different groups, that is the Parkinson's disease and healthy control group. And each of these sample analysis, it was carried out in triplicates. So in the triplicate analysis is provided as the first result, second result, and the third result. In the second graph, the peaks that we are seeing, these are the electrochemical signal that is generated depending upon the chemical composition of the sebum, and the Parkinson's disease is shown in red color, and the healthy control is shown in green color. The places where star is marked or asterisk is marked, these are the places where statistically significant difference are seen between these 2 curves. This shows that indeed there are differences in the signal that is produced from the sebum analyzed from Parkinson's disease patients and healthy control patients, individuals. This plot again shows the difference in the response of SAW sensors among Parkinson's disease patients and healthy controls. Response of SAW sensors per hertz for different time factors, here from 0.8 to 2.7, and here from 5.1 to 10.2. And here this slide describes the area under curve difference for the Parkinson's disease and healthy control. All the asterisks they denote statistically significant difference between the groups. This also confirms that indeed there are differences found between Parkinson's disease and healthy controls in terms of sebum composition. Next, they performed univariate analysis for the area under the curve for Parkinson's disease patients and healthy controls, and with respect to age, in only this case, they have seen that the p-value is less than statistically significant threshold, that is 0.05. In other cases, a statistically significant difference in the p-value was not found. This is the principal component analysis of the volatile organic compounds, and as we can see, for the Parkinson's disease patient, the data points are clustering for the principal components at one particular location, which shows that there is a potential that some differences between the Parkinson's disease patient and healthy controls are present, which can be potentially utilized for distinguishing between the Parkinson's disease patients and the healthy controls. And therefore, the diagnostic utility of the sebum composition can be exploited for clinical diagnosis of the Parkinson's disease. This panel describes the effectiveness of the three classification models that were used, that is the gradient boosting, SG boosting, and random forest, respectively in blue, green, and orange color. So the area under curve, this shows the accuracy and sensitivity and specificity of these three models. So the highest was found for gradient boosting model, where the AUC it was found to be 0.89. This result, it shows the comparison of the electrical signal of the sebum samples in this is stage wise stratification. So here they tried whether the differences between the healthy controls and the Parkinson's disease sebum composition, does it depend on the progression of the disease. But as we can see in this particular result, there is now a significant difference when early stage individuals and the late stage individuals were compared. So the practical utility of this particular method is only limited now for detection of Parkinson's disease versus healthy control, but Parkinson's disease progression cannot be tracked by using this particular method. Discussion. This study, it evaluated whether this olfactory artificial intelligence system, it can be used for clinical diagnosis of the Parkinson's disease. And the gradient boosting model, it showed the best ability to differentiate Parkinson's disease patient from healthy controls. And here the sensitivity of 83.33% and specificity of 84% with area under the curve of 0.893 was found for GBDT model. So the ability of the artificial intelligence olfactory-like system to predict Parkinson's disease progression in terms of the Hoehn-Yahr stage, it was also evaluated, but there was no success in that particular analysis. The results indicate that the artificial intelligence olfactory-like system, it can be a valuable ancillary tool for the diagnosis of the Parkinson's disease, but not its severity. So it cannot be a primary tool to distinguish between healthy controls and the Parkinson's disease, but it can serve as an accessory tool to increase the confidence in the diagnosis of the Parkinson's disease, though the progression of the Parkinson's disease cannot be tracked by this method. This study also has limitation. The first limitation is that the AIO system is data peak intensities that contain the upregulated compounds in the AIO system data. They were not verified by the DCMS analysis, which should have been done. The difference in the headspace due to different sample size and difference in the sebum secretion between Parkinson's disease patient and healthy controls was there, and therefore due to this difference, this comparison cannot be an accurate comparison between these two groups. Again, the third limitation, it was that the number of drug naive patients, that is the patients which did not receive any Parkinson's disease treatment drugs, it was very small. That is only 3 patients were falling in this particular category, and therefore all the Parkinson's disease patients were included irrespective of their drug treatment. And due to this limitation, there might be an impact of the medication that is used to treat Parkinson's disease onto the condition of Parkinson's disease, and as well as the sebum composition of the Parkinson's disease patients. So therefore, if you want to actually develop a diagnostic tool then Parkinson's disease patients which were never given any Parkinson's disease treatment drugs would be an ideal group to compare with the healthy controls. But this study has this particular limitation. In conclusion, a new fast, low cost and non-invasive method it has been employed to analyze the sebum volatile organic compound compositions, which shows that this particular method has the ability to distinguish Parkinson's disease patients from the healthy control group. The impact of the research is that the present study demonstrates that the use of sebum volatile organic compounds based artificial intelligence olfactory-like system, it may be a valuable and clinically accessible auxiliary tool for the rapid screen of Parkinson's disease potential patients. The results of this study demonstrate the promising potential of application value of the artificial intelligence olfactory-like system in the screening and diagnosis of the Parkinson's disease. Thank you for patiently listening. If you have any questions, you can ask in the chat box.
Thank you, sir, for the presentation. Audience, now the floor is open. If you have any questions, please type them in the chat box. Okay, I don't see any questions. With this, I think we can conclude the session. Thank you, sir, for the presentation, and I thank everyone for attending the session.